Hi, welcome back to LPHumananatomika. In the previous session, we have seen the planes of section of the cell and the luminal structure and the appearance of them in different planes of sections. With this, we now will start the discussion of the epithelium. Epithelium is comprised of cells which cover the external surfaces of the body, underline both the internal and the closed cavities of the body and those body tubes which communicate with the exterior like alimentary, respiratory and genitourinary tracts. Epithelium also forms the secretory portion called parenchyma of glands and their ducts and the receptors of certain sensory organs. The characteristic features of epithelium are the constituent cells are closely packed together, the intercellular substance between the cells is reduced to minimum, the cells rest on the basement membrane which is made mostly of amorphous substance. Epithelial tissue does not have blood supply, hence called avascular, and the nutrition is derived from diffusion from the capillaries supplying the nearest surrounding tissues. The surface cells are destroyed by wear and tear and are replaced by new cells produced by mitosis. This phenomenon of destruction and replacement of cells is known as turnover. A given epithelium may serve one or more functions depending upon the cell types present. The epithelium covers the entire external and internal surfaces of the body and forms a barrier. It may be an almost impervious barrier as in the epidermis of skin and urinary bladder. Anything that enters or exits the body has to cross this barrier. The epithelium of the skin protects the body from mechanical injury and invasion of microorganisms. It also prevents excessive loss of water. Any substance that enters the body or is discharged from the body as a waste product must literally pass through the epithelial cells and not between them. The epithelium of the intestine is involved in absorption of water and digested food from the lumen of the intestine. Epithelium may be secretory as in the stomach. The secretory cells of all glands are derived from epithelium. These secretory cells synthesize and release secretory products. The epithelium permits the passage of certain substances through the epithelial layer. For example, gaseous exchange between alveolar air and the blood in the pulmonary capillaries occurs because the epithelia lining them allow the passages of gases. Specialized epithelial tissue in sense organs, innervated by sensory nerve endings, helps in sensory perception. Epithelia of the kidney tubules excrete waste products from the body. The epithelial cells of the sweat glands excrete sweat from the body. Lubrication is observed in serous cavities like pleural, peritoneal or pericardial cavities where a capillary layer of fluid lubricates the interval between parietal and visceral layers and prevents separation of layers. Now, what is epithelium or epithelia? Okay, epithelium is singular, epithelia is plural. Group of cells with the similar structure and functions rest on the basement membrane. We call it as epithelium. It can be one or more layers. If there are more layers, then only the basal layer will rest on the basement membrane. So what is basement membrane? Basement membranes are thin sheets of extracellular matrix that form the supporting structure on which epithelial and endothelial cells rest, and also surround muscle and fat cells and the Schwann cells of peripheral nerves. The basement membrane is composed primarily of glycoproteins, type 4 collagen and laminin that are secreted by the epithelial cells. In multi-layered epithelia, the deepest cells, okay, so here you can see the multi-layered epithelium on the left side, okay, so the deepest cells lie on the basement membrane, okay. So here you can observe only the basal layer is resting on the basement membrane. The other cells are anchored to the adjacent cells, okay. A distinct basement membrane cannot be seen in ametoxylin and eosin preparations, but can be well demonstrated using the special staining method called the periodic acid skiff method. In this method, the glycoproteins present in the membrane are the ones which are stained. Under the electron microscope, a basement membrane is seen to have two parts, a basal lamina which is nearest to the epithelial cells and a reticular lamina or fibroreticular lamina consisting of the reticular tissue and merging into surrounding connective tissue. The basal lamina is further divisible into the lamina densa containing fibrils and the lamina lucida which is transparent. The lamina lucida lies against the cell membranes of the epithelial cells. So, from the epithelium downwards, okay, when we see the layers of the basement membrane, okay, from the epithelium downwards, lamina, the layers which are seen from the epithelium downwards are, the arrangement is lamina lucida, then the lamina densa, okay, both of which are the part of the basal lamina, and finally is the reticular lamina, okay, which is merging with the connective tissue, okay. So finally is the reticular lamina and it binds to the connective tissue, okay. So coming to the functions of the basement membrane, it provides adhesion on one side of epithelial cells and on other side to connective tissue. It acts as a barrier to the diffusion of molecules. The barrier function varies with location. Large proteins are prevented from passing out of blood vessels, but diffusion of gases is allowed on lungs. Recent work suggests that basement membranes may play a role in cell organization as molecules within the membrane interact with receptors on cell surfaces. Substances present in the membrane may influence morphogenesis of cells to which they are attached. The membranes may influence the regeneration of peripheral nerves after injury and may play a role in re-establishment of neuromuscular junctions. In some locations, cells are found aggregated in close opposition with one another, lacking a free surface. These cell aggregates, okay, without a basement membrane are called as epithelial-like or epithelioid tissues. Examples of epithelioid tissues include the intestinal cells of Leydig in testis, the luteal cells of the ovary, the parenchyma of the adrenal gland, and the epithelial reticular cells of the thymus. Now, coming to the morphology of epithelial cells. Okay, let us first discuss the different surfaces of epithelium. Cells have basal surface which is in contact with the basement membrane, an apical surface which is free. In most cases they are covered by coating substances, and a lateral surface which is in contact with the same surface of the neighboring epithelial cells. We will see the specializations of the surface of the cell, okay. So many epithelial cells show projections from the cell surface. The various types of projections are cilia, microvilli and stereocilia. Surface modification of epithelial cells. They help in increasing surface area for absorption in movement of structures and keeping adjacent cells intact. Cilia can be seen with the light microscope as minute hair-like projections from the free surfaces of some epithelial cells. They are motile. The free part of each cilium is called the shaft. The region of the shaft which is attached to the cell surface is called base. Also, it is called as the basal body or basal granule or kinetosome. The free end of the shaft tapers to a tip. Each cilium is 10 micron meter in length and 0.25 micron meter in diameter. Electron microscopic structure shows that the structure at the shaft consists of an outer covering that is formed by cell membrane or the plasma membrane and an inner core called axoneme that is formed by microtubules arranged in a definite manner. There is a central pair of tubules called the central singlet microtubules. This is surrounded by 9 pairs of doublet microtubules or tubules. Okay, the outer doublet tubules are connected to the inner singlet pair of tubules by radial structures which are like the spokes of a wheel called radial spokes. Microtubules in cilia are bound with proteins dynein and nexin. Nexin holds the microtubules together and dynein molecules or arms are present or are responsible for bending the tubules and thereby move the cilia. As the tubules of the shaft are traced towards the tip of the cilium, okay, so here when we see, if we trace the tubules towards the tip, what happens is when we take these doublet tubules, okay, one of the tubule will end in between, okay, they end in between and only one microtubule will reach close to the nearest point of the cilia, okay. So when you trace near the tip of the cilia, we can see one tubule of the each pair of doublet microtubules, okay. So near the tip what happens, this also falls short, okay, close to the tip, and near the tip we can see only the central singlet microtubules, okay. So only two tubules, that is the central singlet microtubules, will be seen near the tip, okay. So at the base of the cilium, okay, at the base of the cilium, what happens is one additional tubule is added to each outer pair so that the 9 outer groups of tubules have 3 tubules each, okay, 3 tubules each, exactly as in the centriole. Coming to the functional significance, the cilia lining the epithelial surface move in coordination with one another, the total effect being that like a wave. As a result, fluid, mucus or small solid objects lying on the epithelium can be moved in a specific direction. Movements of cilia lining the respiratory epithelium help to move secretions in the trachea and bronchi towards the pharynx. In the uterine tube, ciliary action helps in the movement of ova and of spermatozoa through the male genital tract. Flagella are larger processes having the same basic structure as cilia. In the human body, the best example of a flagellum is the tail of the spermatozoa. The movement of the flagella are different from those of the cilia. In the flagellum, movement starts at the base, okay, and the segment nearest to the base bends in one direction, and this is followed by bending of the succeeding segments in opposite directions. Okay, so that a wave-like motion passes down the flagellum. Now coming to the modification, what we will see is microvilli. Okay, they are finger-like non-motile processes or projections from the cell surface measuring 1 or 2 millimeter in length and about 75 to 90 micron meter in diameter. Microvilli are covered by a polysaccharide surface coat called as glycocalyx. Electron microscopic view of each microvillus consists of outer covering of plasma membrane and an cytoplasmic core in which there are numerous microfilaments called actin filaments. The filaments are continuous with actin filaments of the cell cortex. Okay, they are called as cytoskeleton. So the actin filaments are attached to the cytoskeleton, okay, at the periphery of the cell. Numerous enzymes and glycoproteins concerned with absorption have been located in microvilli. Remember, this can be seen in small intestine. Okay. Coming to the functional significance, microvilli greatly increase the surface area of the cell and are therefore seen most typically at sites of active absorption. For example, intestine and the proximal convoluted tubules of the kidney. Okay, so that is the place where these type of epithelia are present with microvilli. Now, stereocilia are another type of the apical cell surface specializations. They are very long, thick microvilli measuring about 5 to 10 micron meter in length. They are non-motile, okay, but the structure is similar to microvilli and the length is almost similar to cilia. Okay, so you can see the length is almost similar to cilia, but structurally it is similar to the microvilli, having an outer covering of plasma membrane, inner cytoplasmic core with actin filaments which are anchored to the cytoskeleton. Okay, since it is non-motile, it is called stereocilia, false cilia. Okay, they are sensory in function and are seen on receptor cells in the internal ear and on the epithelium of epididymis. In internal ear, they are called as kinocilia. Okay, they increase the cell surface area for absorption in epididymis and probably function in signal generation in hair cells of internal ear. Cilia are also present on the cells in the olfactory mucosa of the nose and are called olfactory cilia. They are receptors for smell. Now, we have seen the surface modifications on the apical surface or the surface present towards the apex of the cell. Now we will see the modification of the cell membrane which is present towards the basement membrane. Okay, so in some cells the basal surface of the cell membrane resting on the basement membrane or lateral aspect of the cell shows deep folds which are called as basal folds. Like microvilli, basolateral folds are an adaptation to increase cell surface area. Okay, basal folds are seen in renal tubular cells and in cells lining the ducts of some glands. Lateral folds are seen in absorptive cells lining the gut. If you like the video, hit the like button. If you have not yet subscribed, subscribe to the channel and hit notification button. Join my private Facebook community where like-minded students will support, discuss, encourage and push each other towards reaching the goal. For link, visit the description area. Thank you for watching.